Top of the hour. We're going to go ahead and get started today. Thank you so much for joining us. We're really excited to be here. My name is Kristen Newsel. I'm the Director of Nutrition Programs, and I have here Dr. Chris Mason. Big welcome to you. Thanks for being here.
Hello, Bob. Pleasure to be here. Thanks for having me.
Okay, wonderful. Well, we are going to talk today all about poop. Here at Pendulum, the team of dietitians, we talk about poop all of the time. And I know a lot of times people are a little nervous to talk about this topic, but I will admit I brought it up at a baby shower this weekend that I'm talking all about poop, and people just went right into it. So with that, I will say I'm really excited for this discussion. It's so important for everyone's health to actually talk about poop. So we're going to just jump right in.
All right.
Okay, so just a few housekeeping things. This presentation is being recorded, so no need to worry about not getting it afterwards, or if you have to jump or come in halfway through, we will send these out. So you'll receive automatically an email with the link to the recording in about 24 hours. There's also a really good discount code that we're providing just for joining the Scoop on Poop here at Pendulum. So thank you for attending. This code this time is SCOOP. Easy enough to remember, for 20% off your first month subscription order. And I do want to note that this coupon code is going to expire on this Sunday, August 7th at midnight PST. So make sure you use this coupon before Sunday. Write it down, SCOOP. We get a lot of questions during these Ask Me Anythings, and we're always so excited to answer them, but sometimes we can't get to all of them. So if you do have any additional questions, we really encourage you to reach out to nutrition@pendulumlife.com if you have additional questions throughout here. And I will say for this webinar, if you have questions, if you could just do like Mary Lou did, she put a question in the Q&A. So whether you're on a tablet or your phone or a computer, find the Q&A here in Zoom, and that's how you can submit your questions for today's webinar, not through the chat. And Chris and I will go through them and answer your questions as they come in. Feel free to pop those in as we're talking. Okay, we also have a customer care team member in the chat. In the chat function, it's not working today, but just know he's here and he'll be able to write down anything from the Q&A. And again, this is just a taste of what you'll have access to with Pendulum Probiotics. We aren't just a product, we're a whole support system and want to give you the tools and knowledge you need to be successful to reach your health and wellness goals. By purchasing a membership of Pendulum Probiotics, and I've got a really good code here that I shared with you, you'll also be getting access to webinars like these, free one-on-one coaching with your own microbiome Pendulum dietitian, group classes, and more. So I highly recommend you take advantage of this code that I'll provide at the end, because you'll be able to not only get a great product, but talk to myself or one of the other amazing dietitians. So we're going to go ahead and go to the next slide. So today we're going to talk about how to rate your own poop. What's normal? I get asked that question quite a bit. Your microbiome in poop, and different fiber types and their impact on your stool, Pendulum Probiotics in poop, and then Chris has some amazing slides he's going to share. And I will say, don't feel the need this is a personal topic, so don't feel the need to get into the nitty gritty. If you are a Pendulum member, you'll have access to that one-on-one where you can really dive deep into those more personal questions. I really like to keep this broad. I'm sure you don't want to share too much information. And I hope that you all are not eating a meal right now, because we're going to dig right into, your poop and talk all about the consistency and form, all that good stuff. And why are we talking about this? I like to bring it back. It does tell us so much about our health, and it's a sign of how well your GI system is functioning. So here we have the Bristol stool chart, and I'm not sure if you all have ever seen this before, but this is widely used as a research tool to evaluate the effectiveness of treatments for various diseases of the bowel. And the chart is used to describe the shapes and types of stools. It's also used as a tool to diagnose constipation, diarrhea, and irritable bowel syndrome. So if we go from like the top to the bottom, type 1 has spent the longest time in the bowel, and type 7 has spent the least. A normal stool should be that darker blue there, that type 3 or 4. And depending on the normal bowel habits of the individual, it should be passed once every 1 to 3 days. Some of you might do more, some might do less, but make sure you talk to your doctor about that. Type 1 and 2 may be difficult to pass, and it may indicate some constipation there. So separate hard lumps like nuts or lumpy and sausage-like. In type 3 and 4, these are the ideal stools that I mentioned. Type 5, trending towards diarrhea, and then type 6 and 7 are considered diarrhea. So that mushy consistency, liquid consistency with no solid pieces. And if your poop isn't a perfect, easy, continuous log, it's not necessarily a sign that you're sick, but it may be a sign that you're not eating enough fiber or that your gut microbiome isn't in great shape. Okay, lovely picture of corn here. You all know why we have the corn here, but your microbiome and poop. So healthy gut microbial ecosystem. Here at Pendulum, we are, leaders in microbiome science, and we talk all about the microbiome, but it's generally thought to be characterized by high bacterial richness and diversity. So lots of bacterial strains, different strains, a lot of them, presumed to reflect an ecosystem stability and resilience. And your poop is alive with bacteria, which is kind of crazy to think about. And it's tempting to think of feces as simply the used up remains of the food you ate, the stuff that makes it through after digestion. But in reality, this stuff is present, but about 50 to 80% of your poop, excluding water, is actually bacteria that had been living in your intestines and was then ejected as food passed through. So kind of crazy to think about again, but many of the bacteria in poop are still alive, but some are dead, carcasses of species that bloomed as they fed on the indigestible plant matter you consumed, then shortly died afterward. But it's not all bacteria, so keep that in mind. Your poop also includes some of this indigestible plant matter we're going to get into, like the cellulose in vegetables. That's kind of the corn, the whole corn picture here. Your poop also contains small amounts of your own tissue, so intestinal lining cells that were kind of sloughed off during your digestion. And of course, there's water. And American poop is different than Italian poop, is different than Australian poop. So we might have people from all over the world here today, and everyone's poop is going to be different. It's impacted by your environment. It's impacted by the foods that you eat. There's also poop transplants. And the first time I told my husband about this, now he's a businessman. He's not into the science and poop like I am, but he was so grossed out. But this is actually a fascinating technology if you haven't heard about it. So poop transplants, or FMT as they're called, fecal matter transplants, actually help a lot with health. So you can transplant the stool from a healthy person into a person who might not be so healthy. So maybe they have something called C. diff or things like that, and it can change their microbiome. So the next question I usually get is, how does that happen? How do you give the poop from somebody to the next person? And they can give it through an Ng tube and nasogastric, or as you guessed it, through the other end. That's another way to get that poop in there and help with the microbiome. But it all goes to show how important it is to have that healthy microbiome and how powerful it is. All right, so we talked a little bit about fiber, and as a dietitian, I love to also talk about food and fiber and what can make your gut healthy, in addition to probiotics. So it's good to feed those probiotics. You can't have one without the other. So we have 3 different types of fiber. And as you know, the food that you eat plays a huge role in what shapes, pun intended, your stool. So let's start with insoluble fiber. This is the type of fiber that helps speed things up. So if you're a little bit constipated, the foods in this insoluble fiber list are going to be really important for you. It doesn't feed the microbiome, but that doesn't mean that it's not a good fiber to eat. It helps with colon health, it helps with digestion. And some sources are whole wheat flour, wheat bran, nuts, beans, vegetables like cauliflower, lettuce, green beans, and most plant foods actually have both types of fiber. So going over to the resistant starch, these are carbs that resist digestion and function similarly to fiber. And these actually do feed the gut microbiome. So benefits are they help with digestion, they help slow the absorption of sugar. And some examples are oats, beans, legumes, cooked and then cooled potatoes, and rice. Now moving into the soluble fiber. So think soluble slows things down. So if you have diarrhea and you're on the other end of the spectrum, soluble fiber can help with this. It dissolves in water and forms a gel-like material. It also can help with lowering cholesterol, slowing the absorption of sugar. And some sources are oats and barley, peas, beans, fruits and veggies. Psyllium's a great source of soluble fiber. So whenever I recommend increasing fiber to help with your microbiome, I always say to increase the fluid that you're consuming too, because that could, if you don't, that could lead to, maybe more constipation or more issues down the road. So an added bonus of resistant starch and soluble fiber, as I mentioned, is that they both feed the gut microbiome. Okay, so finding the right Pendulum product for you. As you know here, like I mentioned earlier, we're the leaders in microbiome science, and we have 3 different wonderful products here that I like to talk about that a lot of people ask, which is right for me. So we have Pendulum Glucose Control. So think about your health goals. What are you wanting out of these? Managing type 2 diabetes. This is the first and only medical probiotic designed to help manage A1C levels and post-meal sugar spikes. So if you're someone who's like, oh, I really want to, help my blood sugar levels, then this is the right one for you. Acromensia, this is a really popular one. Everyone loves Acromensia. This is for overall gut health. If that's your goal, then this is the right one for you. It's an exclusive formulation that produces organic compounds to keep the gut and body in the best shape possible. And then GI repair has Clostridium butyricum. So this is to help relieve. This is for occasional gut discomfort. Exclusive formulation includes a butyrate producing strain. So butyrate, you might not be familiar with that word, but that's something that, is really important when you're looking at the microbiome. It's a short chain fatty acid. So it helps with the relief of gas, bloating, diarrhea, and constipation. So before I hand it over to Chris to talk all things that he has, I want to remind, so anyone who jumped on a little bit later, this is being recorded. So we'll be sure to send it to you afterwards. Remember to write this discount code down right now, SCOOP, for 20% off your first month subscription. And you do get access to the Pendulum Dietitian with that. And then if you have additional questions, feel free to reach out to nutrition@pendulumlife.com. I do see some questions coming through the Q&A. So if you popped in late, please continue to ask those. We'll answer them at the end. All right. So with that, Chris, I'm going to go ahead and pass the baton to you.
Unmute. So thank you very much. And that was, Kristen, that was actually a fabulous intro to stool texture catalogs. Also, some of the Bristol scales, everything done with fiber. So there's 10,000 things I could probably talk about. You've covered probably half of my favorite subjects already. FMTs in particular are an extraordinary ability to, transplant an ecosystem of someone and give them better health. Before anyone asks, does it look like I'm in my grandma's house with the butterflies and the birds? We just moved to a new house, and this is where I'm currently set up. We're still unpacking. So this is the quietest room I could jump into for this seminar.
I think it's really your office. It's probably really your office.
Well, currently it is. It's good you can't see the floor, but I'm still we're still unpacking, but we've made it into the new house and wonderful in Brooklyn. So, but I want to chat for about maybe 10 minutes about just some work in the lab that deals with kind of the things we've seen, other adventures, basically. So let me share my screen, and I will keep it brief. Here we go. So So we'll just call it Stool Adventures, and I walk you through some interesting things that have happened that we're working on in the lab, and then we go. I think after my talk, then we'll do kind of the AMA Q&A. If that's right, Kristen, I think, right? Yeah, great.
Yes, that would
okay,
be just perfect.
Great. Okay. So, Kristen asked for the slides politely weeks ago, and I didn't. I just finished them like 5 minutes before this talk. So, in classic, crazy professor style, I just show up with slides that no one has approved nor seen, nor has any idea what I'll talk about. So, first, before I do have some conflicts of interest, including I'm an advisor to Pendulum, but I'm very grateful and happy to be an advisor to such a great team. I've seen them from the very early days of the company to where they are now, with a great manufacturing facility, an expanded team, wonderful people there. So, but I have other conflicts of interest as well. And so I'm going to start with what we've been looking at for Stool Adventures includes what happens even when you go up into space and what happens up there when you're trying to collect samples in microgravity. This is some pictures from Scott Kelly's time in space, for example. And this is other good picture. You can see here some the night sky, and you can see also the very thin, sort of crisp, thin layer of atmosphere on our planet. And so we've had this fortune to work with NASA and other space agencies on some of these questions of what happens in space. And I'll come back to Stool, just hang in there. But a lot of the work in our lab, we do clinical trials on FMTs, we do clinical trials for microbiome research, a lot of work in the R&D space, but also we try to understand what happens when you're in the most extreme of environments. You can see here there's floating microbes. This is one of the key risks for spaceflight that NASA has identified. And we want to try and address a lot of these risks as best we can for current missions, as well as someday eventually going to Mars. And a lot of you probably saw The Martian, or maybe you could grow potatoes out of your own stool, is something that could be in the works. But before we get there, we actually want to, and have been continuing to, profile astronauts at all these different omics layers, looking at the genetic and molecular data. But also you can see one of the functions on here is called the microbiome over here on the left. And because we know this changes in response to diet, in response to disease, but also in response to spaceflight. So something we've built out over the years is something called the Standard Omics Measures for Astronauts, or SOMA. And included in this is collections of stool before, during, and after spaceflight. And also we use some of the same protocols for controls, and also in some cases for clinical trials. So I want to walk you through a couple of adventures that we've done, and then also talk about a new way to collect stool samples that we've partnered with Pendulum on for some upcoming clinical work and in general for customers. So the first little vignette I'll give you was that we recently worked with SpaceX and Trish and others for the Inspiration 4 mission, which was the crew heading up the first all-civilian crew that included someone with a prosthetic limb, and Haley Arsenault on the right, who's a cancer survivor, and what are otherwise regularly more normal people going up into space. So they made their way up into the upper atmosphere, went up into space. They were all very excited. They flew up to higher altitude than the ISS or Hubble, so went really high up into flight. You can see the 575 actually got up to about 590. And then they made it up there and were floating around. So they're very excited. They're very happy. As the mission went well. They came back down after a few days and splashed down. And then we did SpaceX crew picked them up. They were excited and cheering and all happy because they didn't die on the way down. It's always exciting when that happens. And we were excited celebrating near Cape Canaveral because we were going to get blood samples and stool samples. And we were also happy that they didn't die, of course. So it was very good celebrating. And some postdocs that went to collect samples from the capsule as it was coming back towards the launch pad. And we had some interesting questions about, first, with the skin microbiome. And because we know that, for example, skin microbiomes between opposing teams can start to look more like each other if they just bump into each other. So even just during a game of roller derby, it's been shown that you can start to get more similar to your opponents. But we had a really unique occurrence in this mission where the toilet had an emergency alarm during the mission up in flight. And people did not reveal a lot of details, but we had some internal documents and some details that there were no specific issues, but this became a much more intriguing accidental microbiome study that we never intended, never could have ever planned for this, but suddenly had a distinct version of a microbiome study that involved skin and anything else that might be floating around. And though they didn't have a bad dilemma, as reported by People magazine, there are other missions that had fared much worse. There are sometimes pieces of waste floating through the cabin. Some astronauts, such as Peggy Whitson, call these little floating presents. And the challenge is sometimes you can't tell if it's a piece of food or a piece of stool, and they are just not sure. But in other cases, we have advanced beyond the technology for stool collection in space than in the 60s, which was just plastic bags and tape. So we've advanced past that. I'll come back to that subject because we've advanced in other ways as well. In particular, for this mission, we've been swabbing, across different areas of the body, including stool samples. And also we could see that if you look at what happened to their body sites and where we saw more or less diversity, you can see actually we saw a lot of diversity in the armpit. And on the right, you can see the beta diversity, which is how different are they from each other. And then because it went down, that means they became more similar to each other actually in their stool samples than before the mission. So they actually all began on their skin and their stool to be a little bit more like each other during just, of course, a few days in space. And when we looked at very specific species that change, you can see here, this is the differences. Let me zoom in. Some of them were things like Caulobacter, which are gram-negative allogotrophic that even show up in lakes and streams, which is kind of surprising. But we saw Caulobacter and other bacteria that did change during this mission. And we started to look at the HEPA filters even. If you zoom in, you can actually see different microbes that were floating through the space capsule, in some cases from all parts of the body. So we're still doing work on this as we speak. So that was one little vignette. The second vignette I want to tell you, except to say that things can float in space. That's not surprising. Excuse me. Is the other thing is, we're for the next mission wanted to make it a little easier. So they had to collect stool samples using like a little spoon and a scooper, which I think a lot of you on the call, if you're on this call, you're probably familiar with either the hat or the hammock or the bucket or the slingshot, or you call it the ricochet machine. It depends on who you are and how excited you are to try and direct your stool. But we worked, actually for the last few years on getting just an easier method. It's basically just a wipe. So once you've done your business, you can actually just take a wipe, which you normally do anyway, and then put this into a medium that would actually dissolve it. So we just published this paper earlier this year and produced this Full Conflict Vendors. Is a company I was a co-founder of, is the Longevity Thorn Wipe, but you can put it in the container and then it dissolves on the way to the lab. But one of the first questions we had was a new method to collect stool. Does this work better or the same as, say, freezing it or keeping it at room temperature, or compared to, say, Zymo has some preservatives, DNA, RNA shield? And we use this DES, which is dimethyl sulfoxide, and this is ethylene amidrotic acetic acid, and then sodium chloride is what we used here to see how well it would do. And we're actually, glad to see that for taxonomic assignment, so what species are present in your gut, we see very similar patterns across the samples for log abundance, the amount of the samples. And the lower abundant taxa are the noisier ones, as we've seen, but it's actually very similar to intra-replicate comparability. So it's just good to see that the DES samples or the wipe is doing just about as good as Zymo, and better than room temperature. Negative means nothing was done to preserve it. And you can see these clustering, these are the numbers higher, means it's more correlated. And we can see that it was actually quite correlated. And then we also wanted to see, if you look at the positive control versus DES or versus Zymo, we see the very, very similar in terms of what's the abundance of any given microbe that you take. So we see that they're very correlated. You look like it's the same on both axes. Another good sign that we're picking up the same species generally again and again with either of these methods. And then also, if you look at, we also calculate what are called virtual metabolites in the platform, and we could see here that this is what's called the gut biotest, and I'll show you one vignette of that in a second. But you can see here we're getting actually even better than the positive controls in freezing. We're getting really good correlation of a predicted metabolic function of the gut microbiome in this case. And finally, we all want to look at the consistency of itself of diversity. Again, this is diversity. Here you want it to be low diversity because it's consistent. And the positive control and the DES were closer than some of the other methods for preservation and no preservation. So that was also good to see. So if you zoom into all the different metabolites that are predicted, you can see this is just each one of them, say metrics for cholesterol, butyrate, calprotectin, constipation risk, taurine, creatinine, so some of the different vitamins on the top here. So you can see for the most part, very, very correlated across all these commercially available preservatives. So with that done, we're kind of excited that it's now launched. And actually, quickly, this is being recorded, so I want to quickly show also what does it look like when you have an account. And this is, if you get feedback from sequencing the data, this, for example, is my account here, and you can see what is my Acromensia. I have very high levels of Acromensia, so I'm very excited. But there's only one place you can actually get this species. You either have to have it natively or you get it from Pendulum. So I'm very fortunate that I'm doing okay. But there's other keystone species that we keep track of when we do the sequencing. And this is the one that at Thorn and Longevity, we recommend immediately say, okay, if it's low, there's really, like I said, only one place you can get it. So this is just an example of other keystone species that we keep an eye on as well, that I just showed you some examples of here. Okay, so what we're doing now, just to keep it into a short vignette, we're doing all kinds of things in my lab at Cornell and also with partnerships with fabulous industry partners like Pendulum. and thorn and longevity, but we're also doing on an academic side, swabbing other places around the world, including other hospital systems and sewage systems. We're sampling wastewater, which is the more mobile version of stool samples. And this has included a big project called MetaSub, where we look at subways and urban biomes and how they change and move. And so this is looking mostly for COVID, but we also see lots of other species in the wastewater. And we've been doing this now for 7 years, actually just finished this last summer, and continually sampling around the world, looking in urban surfaces as well as in sewage. And we can find actually new viruses all the time from wastewater and sewage and city surfaces. This is some of the papers we published recently, showing that every time you flush the toilet, there's things you're washing away that could be new biology, new viruses. So I like to say these days, every time you flush a toilet, if no one takes a sample, somewhere an epidemiologist cries, because you lose the information, or it used to be lost, but now you can get it when you go to the processing plant and actually collect at least some of the information there. But as you've heard already multiple times, there's a lot of information in your gut. We're also going to be doing more missions. So this is some Axiom missions being planned with Commander Peggy Woodson and pilot John Schaffner. Eventually, we're going to be back up with SpaceX for the Starship, which is going to be the larger rocket that eventually will hopefully take us to Mars. And there'll be a first EVA on a private space flight, hopefully later this year. And it's not going to be the last time. There's going to be 4 different space stations eventually orbiting our planet. They will all have to figure out somebody to have their bathrooms work and collect stool samples. So we're working with actually a lot of them now to look at the better methods, including the wipe for collection. And eventually maybe we get to Mars and collect samples there. I think this is the other thing I'd just say, is that we are hoping to get there. And eventually there'll be health metrics, microbiome collection, and a lot of work to be done there. And since I'm at time, I'll just quickly say, we're not the only ones. China's trying to get to Mars, and Mars is not the only planet. There's also several thousand exoplanets outside of our solar system, which in theory we could go. And so there's nothing to do with stool, except that planets, when they're very small, look like the higher numbers on the Bristol stool scale, because they look like pellets. And then eventually we could get to planets that are similar to Earth. You can see purple and blue planets are similar to the Earth Similarity Index. And if you look at it within 15 parsecs of our sun, we can see there's a lot of other planets that actually could be habitable even today. And in 20 parsecs, there's even more. And if you look at the top or side of our galaxy, there's all these planets that we could, in theory, go to. And along the way, we'll have to good gut health is my only real connection there at the end. But I want to thank everyone. It's a shameless book plug, because I just wrote about this. And thanks to the funding from all these different providers for the Cornell lab. Thanks to everyone in the lab who makes this work possible. It's an amazing team. And thanks to everyone listed here as well, friends and collaborators. And finally, thanks Pendulum. And happy to chat and take some questions.
Perfect. Thank you so much, Chris. I find that wipe that you talked about is just brilliant, because I don't know if anyone here has ever tried one of those. You send in your stool and you want to figure out what your microbiome looks like, but I failed miserably, and I can't be the only one. So this wipe is something I'm really excited about, and I would definitely encourage other people who might fail like myself to try it out.
Yes. Well, and there's buckets, there's hammocks, there's other methods, and there's no dignified way to collect a sample that way. And if someone's knocking on the door, they say, "What are you doing?" You say, "Nothing. There's a private moment and nothing's to see here." So
yeah, definitely. It's definitely a brilliant idea. So we've got some awesome questions here. I'm going to go through them. Let's see here. So we've got one from Charlotte. Can I get enough fiber if I don't eat any grains? Oh, this is a really good question, Charlotte. And a lot of people wonder this, and you absolutely, I do have people who don't eat grains. Now, grains are a great source of fiber, as we talked about, but if you're focusing on your fruits, your vegetables, your nuts, things like that, chia seeds, that would be, something that you could totally do, as opposed to like the whole grain bread that a lot of people think of right off the bat. But you can absolutely, focusing on those foods, it's a lot easier than what people think of. So really great question, Charlotte. Let's see here. Let me check that one off. Let's see. I'm trying to get one for you, Chris.
Yeah, there's a whole bunch of a lot of FMTs. Well, I could jump in on one, the efficacy of fecal transplants. There's actually a great website called thepowerofpoop.com, which was originally kind of a self-help site for FMTs. And for certain indications, that could be as much as 60 or 70% effective. If you're looking at ulcerative colitis, in some cases, they've been there. In the best case scenario, they've been that successful. So for other diseases, if it's just IBS, or IBD, it won't be as high, but we kept seeing them be pretty extraordinarily successful. And actually, there is, several nonprofits that will help with this. There's also some clinical trials. Some are at New York Presbyterian where you could enroll for transplants. And so, Open Biome is one of them. I'll put it in the chat box here, but also is a way where you can actually look at FMTs and get information about how well they work and providers. So I'll put that in the chat so people could take a look at it.
Well, sorry, but the chat box is not working for some reason. Well, wait, it just went through.
Openbiome.org. If you, yeah, it works for me, I guess. Okay, but that's one place I'd recommend.
Okay, let's see here. I'll try putting it in there and see if anyone gets this. .org. Perfect. Okay, thank you so much. All right, so we've got a question here from Kimberly. Will Acromension or any of your other products help with SIBO? Thank you. So we don't know that answer. We do have quite a few customers who are taking our products who have SIBO, but we really refer them back to their physician. So I recommend, Kimberly, that you talk with your physician to see if any of these are right for you, because it's going to vary from customer to customer. And I know it's really frustrating when we don't have data to back up an answer like this, but I just like to be completely honest that, hey, you know what, we don't have data on this, but we do have customers who do have SIBO and are taking this. So definitely talk to your doctor about that one. All right, Chris, you see anything in there that is up your alley?
Yeah, actually, I see one. Any intel on how answer live on how the microbiome group has been modified thanks to the COVID virus? Click that one. So actually, we published a paper that showed people who get COVID have a microbiome disruption, at least in the nasopharyngeal area. We can see it there, and then also for long COVID, people with long-term symptoms, we also see disruptions in the stool microbiome. And so it seems to be a systemic effect. We get it in both places. I'll put one of the papers that we published on this that showed, started to show this. It really is not surprisingly a disruptive infection, but also even in places where you might have not originally thought, like in your stool microbiome. Or basically, what it normally does is takes what are some of the normal keystone species, kind of your normal flora, your natural flora, which are commonly present and when you're healthy, and a lot of them get really ablated and are kind of lost. And so it seems to be it's either creating such a disruption at the site of infection that they begin to change, or the immune system is responding. There's also some evidence that, though this is early, that people who are already walking around missing some of these normal flora maybe at a higher risk of infection because it could even be something protected. That's very speculative, but there's a lot of discussion about what's the mechanism. Like, did the virus come in and cause this disruption, or were you already disrupted from something else, say antibiotics earlier, and then the virus had an easier way to get in? And so I think it's not clear, but it's being debated a lot in the literature and at conferences as to how much, which one came first and what's the mechanism driving it. But in general, I put one paper in where we did look at some of those questions.
Thank you, Chris. Such a great questions. We've got a lot here to go through. So I've got one from Jason. Do you get the membership and access to a nutritionist with a recurring purchase of any of the 3 products? The answer is yes. We here at Pendulum, we really want to provide you support. We're not just a product, we're a whole program. We want to provide you with that support to help nourish your microbiome, along with the probiotic and the appropriate eating plan. We say eating plan, not diet, because I promise you we're a group of really fun dietitians, not scary. So yes, you absolutely do receive the free one-on-one coaching. We also have free group sessions after that. So if you want that group session, you can totally join and have a little support system there too. So be sure to take advantage of that coupon code I mentioned earlier, SCOOP, if you haven't already. All right, Chris, I see another one here from Alexander. Is FMT legal in the States and Canada, or still just authorized for C. diff patients? If yes, any recommendations?
It is often through clinical trial. It's not. So there's a few companies that are trying to take it so it become an actual automated product that hasn't succeeded yet. But it's not illegal. You can take it, just have to do it in the context of a clinical trial through physician care. It is not yet a product, though, that has been so well perfected that you can have just one pill that's the consistent ecosystem that you always get. That is what some would consider a holy grail of stool transplants, is that you could have something that works, even if it works 80% of the time for people you'd give it to, that would be phenomenal. There are other people, though, that are often called super poopers, people that have the kind of the best donor profile, which happens for normal organs too. Think of like a liver or kidney. You have to find the best match, right? So this is all very logical to people today, is that you look at the immune profile of the donor and the recipient. Ideally, if you have an identical twin, that's even better. You don't even have to take immunosuppressants in some cases. You can just, basically, you basically have a clone of yourself walking around that you could grab an organ from. Although you should ask first, right? But basically, we don't quite have that yet for stool, where we have a perfect matched, a healthy version of your microbiome before you had some disruption or disease. But the FMT companies, they're legal, and they're working on ways to get them more robust and more essentially identical between the different donors, and also to do subgroups of types of donors. So it's ongoing work, but there are and more clinical trials happening to try and get them into phase 2 and phase 3 clinical trials.
Super poopers. I am absolutely using that term moving
Super
forward.
pooper, people that are, and now everybody wants to be one. If you don't want to be like a sad pooper, no one wants to be that. That's very, it's upsetting.
Yeah. Definitely want to be a super pooper. That's fantastic. Okay, we've got a question here from Yolanda. Can you take pendulum glucose control with metformin? The answer is yes. We get asked this a lot, and actually in our clinical trials, the participants actually were taking metformin along with pendulum glucose control. So that is totally fine to take together. All right, got one little comment here from Carol. It was an inspiring documentary on this SpaceX flight. So cool.
And where was I trying to pull up the question? SpaceX. Well, in terms of what's really interesting, actually, it was a great crew. The thing that was great about them is it was an all civilian crew. They, really didn't just want to go up to space just, for giggles and grins, but they really want to contribute to science as much as possible. And the commander for the mission is Jared Isaacman. Really insisted on, we're going to be up there for a limited time, but how much science can we pack in? What can we do? So, they did swabs, and we had stool, urine collection, saliva, took, quite literally blood, sweat, and tears, because we have some tear collection methods as well. And they, wanted to look at host response, like so the human cells change, but as well as all the microbial changes in skin, stool. Also in urine, you can actually see, which tissues in the body have died most frequently, and then to come out in the urine in what's called an epigenetic analysis, meaning that there's different chemical marks on each piece of DNA that comes out of your body, whether it's human or microbial, and we can see often where it came from in your body. So this is, ongoing work that we're doing with the crew, and we're monitoring the microbiome of them between each of the missions as well, because Jared's actually going to go up for 3 more flights, including the Polaris Dawn mission. So there's actually a really unique opportunity to see what happens to someone's microbiome over many years. And there was one other person, Eric Alm, who did this at MIT. He kept sequencing his stool about once a week for about 7 years, last I saw his chart. And you could see when he had diarrhea, for example, because he was traveling abroad and suddenly got an infusion of some less friendly bacteria, or at least just much different. So there's very clear tracking. If someday, I'm hoping we could all have a device connected to our toilet that would give us a report back every day or two on how we're doing. We're not quite there yet, but this smart toilet will be sometime hopefully deployed in our lifetimes.
My husband would absolutely buy that. He would.
Yeah, I think even if it was, you would have to pay, because the reagents would have a cost. So if you paid, there's something just mentally taxing about thinking that you're paying like $20 just to go to the bathroom if it costs that to process it. But maybe some people would do it. I think
people would do that. I think absolutely they would.
Yeah, if it came with like a chocolate or something, I think that would be white chocolate, so it'd be differentiating.
There you go. I like that. So we have a really good question here from Claire. How does food restrictions impact gut health? So food restrictions and different restrictive eating plans can really impact your gut health in a lot of different ways. It really just depends on what you are specifically restricting. It can alter the bacteria ratio in your gut microbiome. It can alter your pooping habits. So it really depends on what you're restricting, specifically fiber. and probiotics. So if you are restricting fibrous foods, that's going to decrease the density, so how much of certain strains, and also eventually, because they're synergistic, the diversity of your microbiome. And if you're not, taking probiotics, or if you've lost them somehow over time due to your environment, stress, medications, you're going to need to supplement with probiotics. So restrictive diets can absolutely alter your gut health. So it really just depends on, like I said, specifically what you're taking out of the eating plan. All right.
One of the questions is what percentage of people have lower amounts of Acromension naturally in their gut, lower or higher? I don't know. I just sent a quick note though to an analyst at our team. I'll see if I can get that answer before the hour is up. The kind of the distribution of how often do people have it, because we could look in our own database and find out. So I remember it being relatively kind of like 20-25% of people that had it on the high level. So it was fairly uncommon to have it that high. But I will find out. I'll see if I can get that chart during the Q&A.
Perfect. Thank you. I've got a question here from Heather. When is a good time to take probiotics? So specifically for Pendulum probiotics, for Pendulum glucose control, we recommend twice a day, 1 capsule twice a day with food. So 1 in the morning, 1 in the evening. If you just want to pop it in the refrigerator, maybe buy your morning beverage to remember. I always like to remind people to do that. With Acromension and GI repair, it's 1 capsule once daily with food. So that's when it's recommended. Really good question. All right, Chris, I see another one about FMT in a follow-up. Could an FMT eliminate SIBO, or is this outside its realm of effect?
It could, especially a lot of times before you do FMT, you do a really strong push for antibiotics and try to clean out what's there. It's possible. I think we've seen, this is why the field has taken so long to get robust, consistently made ecosystems in one pill that work for everybody. In some cases, it seems to work amazingly well and people seem to be cured. In other cases, people go back for 2 or 3 or 4 FMTs and it's not clear why it's not taking hold. There's a lot of discussion about the structure of the ecosystem, and I showed those keystone species before because I wanted to point out that it's not just Acromension, although it's one of the most key factors you could have for microbiome health. But there's other species, of course, that kind of make the house of your gut health work. Or thinking about having pillars of a house, and you need to ideally have 5 or 6 really strong, pillars to hold something up. And Acromension is one of them, but ideally you have all of them in really good condition. So I think in some cases it's the missing microbes, but in other cases people have very different immune systems and immune function. And your immune function also decreases as you age, what's just called immunosenescence, meaning we all just slowly become less functional as we age. We may become more interesting or peculiar as we age, but we immunologically become less useful generally, and cognitively, and somatic mutations. And generally, we just fall apart as we age. But along the way, you get lots of wisdom. You can have a lot more Saturday afternoon coffees and play cards in the park. There's a lot of other advantages to getting older. But the immune system changes a lot. That's the end of my soliloquy on aging. But the immune system changes as well, and you have to take that into consideration as you get older.
Great. Thank you. We have another really good question. I blend vegetables daily. I also eat beans, which I soak them and pressure cook daily. Will this cause any gas? So anytime you have fiber, it could cause gas. What happens is whenever you eat fiber, it goes into your gut, into the large intestine, and the bacteria that you have in there kind of breaks it down and it produces short chain fatty acids, which we want. This is for our health. We absolutely want this. And gas can occur then. So what I recommend when people are starting to add more fiber to their eating plans, I recommend going slowly to help sort of your body adjust. If you're someone who doesn't eat any fiber, you want to do this slowly over time. So that way we can sort of prevent it. But that's totally normal. Usually your body acclimates if you do it gradually over time. But yeah, it's just part of our bodies and how we digest things. And some people are gassier than others, as we know.
So one thing I also mentioned is there's several short chain fatty acids that you want to keep an eye on, and so as Kristen just correctly noted, this is produced a lot by the microbes in particular. You'll get it from Clostridial clusters of species, but butyrate, lactate, propionate. These are just some of the known short chain fatty acids in stool. And so there's just another example of a chart that we make of tracking these different short chain fatty acids. And so, and there's also different kinds of fiber that will be processed in different ways by your gut microbiome. And so, but this just shows you an example of some of the ones that we keep an eye on, because they all can influence some of your general health, inflammation, and also the changes in your gut.
I love that you have that on there. I was just listening to a webinar about how a lot of people are really focusing more on looking at short chain fatty acids as opposed to, some of the microbes, and how that's such a big indicator for health. So yeah, that's great. And we've got a lot of butyrate producers at Pendulum Probiotics. So
Yeah.
let's see here. I'm already a Pendulum subscriber, Acromension, and I didn't realize I could contact a nutritionist. I do have some questions. How do I contact one? Thank you. So Carrie, I'm going to take note of this. If you want to pop in your email address that you use to purchase Pendulum, I'll go ahead and shoot you an email after this. I'll be happy to. And anyone else, feel free to do so as well, if you're interested, just to get that support.
And how can I tell? Let's see. Oh, here's actually one interesting. If someone has no detectable Acromension, conceding it for a few months along with probiotics, I mean, they could stop someone and keep culture with proper fibers. You see that question? The is basically, if you don't have it, could you? So it's actually a fundamental question in a lot of microbiome studies is, how sticky are your probiotics? If you give someone a probiotic, will it stay? Like, if from a business perspective, if you are creating products that people no longer need to take, on the one hand, you've done a great job, but on the other hand, you don't need to sell anything anymore, so it's not as good for a business sustainability. But we've seen actually from some of the trials at Pendulum and just in general compared to other probiotics, other species, generally about 10 to 15% of people we'll see can actually maintain a probiotic where it colonizes, it stays, and it'll still be there, say, a year later, 6 months later. It seems to be more the exception than the rule, at least from data that I've seen in our own hands and in published papers. It can happen, but it's less likely from what we've seen. And every species is different, of course, but I think it is something that is uncommon, but not impossible. And the best way to do it is to obviously check your gut health and function, but you could even just sequence what's there. And then you'll be able to tell, oh, is it still there? And you can, some of the charts I've just been showing you, you can quantify it and see that it's there and that it's really been maintained.
Thank you, Chris. Yeah, that's a really great point. Either the transient or the colonizing. We get asked that all the time. Got a question here from Kathy. What should you do if you have to take an antibiotic? So this is something you should really talk to your doctor about, general recommendations. And I have some doctors will recommend a probiotic when you're taking an antibiotic, but research shows that if you introduce a probiotic 24 to 48 hours after stopping the antibiotic, that you have a much better chance of it repopulating and, getting your microbiome back to normal. But definitely talk to your doctor about what they think is best for you. Really good question. All right, Chris, I got one for you from Diana. How do fecal transplants jumpstart good microbiome when taking probiotics may require continuous use? Is there some sort of symbiotic relationship among all the healthy microbes in fecal transplant that cannot be achieved by taking individual probiotics?
Yeah, great question. And indeed, there is a synergistic and ecosystem kind of effect. It's much like taking, say, a single plant out of a rainforest and saying, okay, if I put this in my backyard, do I have a rainforest in my backyard? Right. So of course we'd all say, well, no, you need the whole rainforest. You need this really large, complex panoply of species that are all interacting, plus the soil, plus even everything that's in the trees, including animals, plants that are also mosses, insects. Like ideally, you'd want the whole ecosystem to be moved over into your backyard before you could really say I have a rainforest. And the same is true for your gut. You have to, the advantage is you transplant the whole thing. The disadvantage, though, is it could have things you don't need or could be disruptive in their own way. And so it is not just moving a species or a handful of species, it's moving sometimes hundreds of species over at the same time. And so that's both the benefit and the risk is that if it works well, it could be permanent and stay. But if it's not working well, then you've got to get rid of that new ecosystem.
Great, thank you. So we've got another question. I have long COVID. What can I do to restore a healthy and normal microbiome? So That's a great question. However, this is one of those things where we just don't have data, at least not to my knowledge. I don't know if Chris does, but with the microbiome, we're learning so much every single day. We're sort of in like a renaissance period, as one of my colleagues said. And we're learning so much about the microbiome every single day. And in addition, COVID's still fairly new. They're still running, a bunch of studies and trying to figure out all sorts of things about COVID. So Chris, I don't know if you're able to help answer this person.
Yeah, so the long COVID, we're still learning about both, as you just pointed out, is really there's the range of probiotics that are sold by, you could take some Pendulum products. There's a lot of other products on the market, but at this point, just to be totally honest, we don't know yet which ones are the best for long COVID. The best way to tell, would be my guess, would be to take one of the tests I was just showing, where can you look for something that's expected to be there in a normal person, but is completely absent or been disrupted, and then try and tailor what you take to what is missing. Because if you could grab something off the shelf, it might work fine, but ideally it would be much more tailored to what has been lost. And as I was describing earlier, we do see microbiome disruption, consistently in COVID patients, in really severe fashion too. So it's something that we see can persist for a long time. And then long COVID also really interestingly creates this inflammation state that can be around the whole body, which can also affect the GI tract. So as everyone probably knows, it's good to just avoid getting COVID if you can, but even if you do get it, at this point, it's staying healthy as much as you can otherwise. And actually, one of the really peculiar thing, there was a talk just to think about the mechanism for this, is the nucleocapsid protein that's made by the virus can actually sometimes embed in the GI tract. This has been shown from patient autopsies that we looked at. So if you think about your body, as the virus, you think comes in you, it replicates, but eventually your immune system gets rid of it, right? You think, well, maybe it's all gone. Some more recent data from a lab at Rockefeller and some autopsies that we've done, you can see like bits of the proteins, the chunks of the virus are still embedded in parts of the GI tract or in parts of your body. And it takes a while for them to clear out. It doesn't just clear out in a week or two and you're done and there's no trace of the virus left in your body. For long COVID patients, sometimes you can see it persist for months, little bits of the virus sticking around both the protein and the RNA. So, and we're still learning about this. This is really unusual. You don't normally see a virus do this. You don't normally see a virus cause anosmia, the loss of smell and taste. Unprecedented in the history of viruses to see something so focal and causes neurological disruption that then comes back for most people most of the time, but not everyone. So like really unusual things about the virus. And even the tropism of the virus changes. So where it goes in your body, if it's an alpha or delta, it's more in the nose. But if it's Omicron, it shows up in saliva first. Also just kind of weird and interesting, but creepy that the virus is changing kind of where it goes in your body, depending on which strain you've got. So, and all this we're still learning about. So it's a very unusual time. And what it's doing to the microbiome and the gut is for sure, as we know, something, but we're still learning.
Yes, definitely. And as you know, 70% of your immune system is in your gut. So taking probiotics is a great thing. And I've got a question here. I've been having some gut and digestion issues, healthy bowel habits though, and no diabetes, but it is very common in my family. Working with my doctor to figure out what's going on and getting a full GI panel as I travel internationally a lot. Wondering which product is best for overall gut health. Notice that glucose control has the most variety of bacteria. So is that best? Really good question. We get asked this all the time. Which one should I have? It sounds like for you, you are looking for something with the most strains. And so Pendulum glucose control is going to be the one that has the most strains. It's going to have the Acromension. It's going to have the strain that's in GI repair. They're all included in Pendulum glucose control. We have tons of people who take it who do not have diabetes. So it's really up to you on what your goals are. And for you, it sounds like, with that family history, Pendulum glucose control might actually be a good option for you, but definitely something to explore a little more. Okay, let's see here. We've got time for a few more questions. Let's see. I don't know, Chris, can you find one that you would like to answer?
There's some, how can you tell if Efficacy tool support production of erythrin A? There are, I was alluding to this before, is that there's ways you can take the genetic sequence of some of the strains that are in your stool and see what they're likely to produce. one of the best ways to actually confirm it, though, is to do a molecular or mass spec based confirmation of the molecule you're looking for. But some of this can be done with sequence-based methods like the gut bio report here I was showing you. So that's one way to be able to tell that. So that's one question I could see.
Got one here from Bob. How long does it take for the Acromensia probiotic to take effect? Oh my goodness, we get asked this all the time. When am I going to see results? So for Acromensia, my question is, what are you looking for specifically? Why are you taking this? And what are you looking for to see success? It's going to be different for everyone. Everyone's microbiome is starting out in a different place. You might have more Acromensia than someone else who's in this webinar and starting to take it. And so they might see results earlier. So everyone's going to be different, but the difficult thing about Acromensia is that it actually lives in the mucin layer. So it's hard to show up on stool samples. So that's just something to keep in mind, is that it is, kind of trickier to detect just because it likes to live and hang out in that mucin layer as opposed to in the stool like most other strains. So, it's really individualized to where your starting point is and what you're looking for. We don't have an exact answer like, oh yes, at day 25, you're going to just see tons of results. So it's really individualized. But typically with like pendulum glucose control, people start to see results within about 3 months because you're looking at their A1C, which is a measurement of what your average glucose has been over the past 3 months. But people, if they're wearing a CGM, they can see results sooner than that too. But everyone's microbiome is different. Okay, Chris, what would you like to answer here?
See, what's the best fiber or polyphenol to feed your Acromensia? Actually, I don't know. I'm not sure. I don't know, Kristen, if you know that, but that's a good question. Like, do you eat a wicker furniture set, or do you take what you put in your mouth?
You know what? I love that this person even knows what a polyphenol is. So that makes me really excited. So polyphenols, there's over 8,000. And if you look at a list of like the highest amount of polyphenols in a food, you're going to find cloves. But highest amount does not equate to quality. And so I've talked to a lot of physicians who have actually done research on this. There's a lot of research in mice, but I've talked to a lot of physicians who have done this research in humans, and cranberry and pomegranate and Concord grape seem to be the 3 polyphenols that pop up the most that increase your Acromensia. And we actually have a meal plan at Pendulum on our site that is a polyphenol boost meal plan. It focuses a lot on polyphenols and fibers to help with Acromensia. So I do recommend popping that in the cart if you are getting Acromensia.
Let's see, is glucose. And people who are taking antibiotics, I think it just depends on tracking your glucose control. And if you've been on antibiotics, it can disrupt things, and it's good to keep an eye on what your glucose levels look if they're spiking. There'd be evidence that you should take something else. There is one other question: what at-home stool test do you recommend? I have a complete conflict of interest because it's our own test, but it's the one that we built. I built it literally myself, so one I can stand behind and say this is one. It's the one I was showing you of my own my own stool samples, and so it's one that we have been using. So that's one I'd recommend.
I do not have a conflict of interest, and I'm going to say the wipe because of ease of use and efficacy, most likely. So, yeah. Good question. Let's see here. Got time for a few more. Got that email. Great. Considering that every poop is different, is there a chance that Acromensia may not be so good for Brazilians, for example? We don't have that research. Everyone's going to be different. So I recommend, maybe getting that poop wipe and seeing where you're at with things. But we don't necessarily know that Acromensia might not be good for a certain ethnicity or country.
In theory, yeah, there are differences between, as you were saying before, the Italians or American or Canadian microbiomes. They're different, so it's very possible that there's some differences or different strains that you would want, depending on what's normally present. So it's a very good question.
Yes, good question.
One more question. Let's see. I put the link in the chat box for the website.
Yes, and we can send out some of those links in an email if the chat is not functioning properly. And then I've got a question, what is Acromensia for? It's for overall gut health. There's a lot of research out there on, its association with lower weight, better improved metabolic health. We don't have any clinical trials to support that, but that's what's an association for. And so we recommend it for overall gut health. It really helps thicken that mucin layer, which is what we want to do. You don't want to think of your gut like a pipe. You don't want a leaky pipe where things can get through. So Acromensia is kind of like a Pac-Man in your mucin layer. All right, guys, I think we're at time. So thank you so much, Chris, for joining. Always wonderful to hear you speak. I always learn so many new things. Really appreciate you having you on here. And as a reminder for everyone for joining, please feel free to use that coupon code. It is for new customers only. The coupon code SCOOP. Easy to remember. You attended the Scoop on Poop. We've got people's information here to reach out, and we're going to send out a list of some of these questions that didn't get answered. So in the links that Chris kindly put in there. So thank you so much for joining. We hope to chat with you soon, and hope you have a great rest of your day.
Great. Thanks, Chris. And thanks, y'all. Good pleasure to be here.
Thank you. Bye.